Alyssa Martino, PhD student at the University of Sydney. Alyssa, I'm just passing the screen to you if you would like to accept the invitation to present there. Now, Alyssa is originally from Perth and studied agricultural science and genetics at the University of Western Australia before moving to Sydney for her honours in biochemistry. Alyssa will be known to some of you as Myrtle Rust Martino, being active on Twitter under that handle. Those of us working in communications certainly appreciate her efforts to raise awareness of Myrtle Rust. I understand she is a dab hand in the lab as well, and I'm looking forward to her presentation today. Now, are you all good there, Alyssa?
I think so. Can you see my screen and can you hear me?
I can certainly hear you, and there are your slides. They're looking lovely. So
Wonderful.
You should feel free to get underway.
Okay, thank you, Renee, for the introduction. And yeah, thank you everyone for logging in today to hear me give a quick introduction to some of the work I have been doing as part of my PhD. So as Renee mentioned, I am a PhD student at the University of Sydney. I am currently conducting my work under the supervision of Robert Park, Perry Tobias and Brian Jones, and I'll just be introducing some of the work with Melaleuca quinquenervia and how we're working towards using this as a model species for Myrtle Rust research. So for those of you outside of Australia, just a little bit of a background into the impact that the pathogen is having in Australia. So since it arrived in Australia in 2010, it has spread to all of our states and territories aside from South Australia. However, some spores have been detected in traps in South Australia, so we are keeping a watchful eye of what's happening in that southernmost region of Australia as well. Unfortunately, recent climatic conditions here on the East Coast have been very favourable to the pathogen, and as a result, myself and other colleagues have seen an increasing number of disease symptoms on a lot of our tree species over here. So on the right here, we just have an example of the broadleaf paperbark, so the Melaleuca quinquenervia, and you can see it's very heavily infected with Myrtle Rust here. What's really interesting is that we are seeing variability in response to the pathogen out in the native environment as well. And as an example, the tree that was sitting right next to this one was showing no symptoms at all. And so it really piques our interest as to what's going on genetically to be leading to this variability in response. I guess very early on in my PhD journey, when I was just starting out and looking at the literature, something I noticed was that a lot of the research is spanning across a lot of different host species. And so part of my work has really been to use a single host species to refine some methodologies to be able to study more the genetic basis of resistance to Myrtle Rust. So to do this, I have been working with Melaleuca quinquenervia, which is the broadleaf paperbark. It's a keystone species here on the east coast of Australia, and it has some very important ecological roles and also some important economic roles as well. Why we chose to work with Melaleuca quinquenervia was firstly because it can be fairly easily propagated from cuttings and very easily grown from seed. The seed is also available all year round, so there's no limitations in going out and collecting material from these trees. The seeds also germinate very easily, so they don't require any specific stratification requirements, so it makes it really easy to work with. Importantly, for any genetic studies, what we see is that the species shows a variable response to Myrtle Rust infection. So we see some hosts that are highly susceptible all the way through to highly resistant as well. So the main aims of the work I have been doing in conjunction with some collaborators is firstly, we were interested in assembling a high quality genome of Melaleuca quinquenervia, interested in annotating the repertoire of resistance genes, so doing this with the genome as our basis, and then identifying some specific genes and pathways of resistance within Melaleuca quinquenervia. This will then be expanded to other Melaleuca species by screening them for their response to the pathogen. And the hope is that we can take what we learn from the Melaleuca quinquenervia and interrogate these other Melaleuca species that we've screened for genes that we have identified in Melaleuca quinquenervia in response to the pathogen. So first off, just a bit of a summary of the work that we have conducted in assembling a phased genome of Melaleuca quinquenervia. So for anyone that is working in a genomic space and anyone that's not, I'll try and keep this simple. But for most genomes, they are presented in a collapsed form. And what I mean by this is that we have, for example, a diploid organism such as Melaleuca quinquenervia with two sets of chromosomes, one from each of the parents. Generally, when genomes are assembled, this will be collapsed into a consensus sequence. So we just end up with one sequence for the whole genome. As opposed to phasing, this allows us to actually separate the two parental genomes. So we have haplotype A, which is from parent A, and haplotype B, which is from parent B. A couple of reasons why this is important for us in our studies of Myrtle Rust. So firstly, what I will say is that the genome that has been assembled has been assembled from a tree that is resistant to Myrtle Rust and controlled inoculations. And we really hope to use this genome to aid in the identification of resistance genes. We could do this, and this has been done in collapsed genomes, but where a phased genome is more powerful is that it actually allows us to analyse the allelic variation in highly polymorphic gene families. And that is the case with the family of genes that we are looking at. So some collaborators at the Australian National University and my supervisor, Perry, were involved in developing a pipeline called the Find Plant Analyses pipeline. And this was developed to identify the Nucleotide-binding Leucine-rich Repeat genes, or the NLR genes. And it was developed to identify them in Melaleuca quinquenervia, but it can really be used for any other plant genomes as well. So for anyone that's not familiar with these genes, it is a family of resistance genes encoding proteins for pathogen, for detecting pathogen effectors, and it also mediates the host response to those pathogens as well. They are modular in their domains, so we have a variable and terminal domain, which you can see here. So this is generally a coil-coil domain or a toll interleukin receptor domain. And this region is particularly important for pathogen recognition and also signaling. And then we have the central NB-ARC domain, which is highly conserved among plants. And this is what allows us to interrogate the genome for these genes. So this is, it has several roles, but one of the important roles is the activation of the hypersensitive response, so that localized cell death that we see in resistant species. And we also have this leucine-rich repeat region at the C-terminal domain, which is highly variable in their sequences, and it's important in mediating protein-protein interactions. We also see in some these novel integrated domains here, and I'll go into a little bit more detail about the role of these integrated domains after. And we also see some that don't contain this N-terminal domain. So these genes are highly polymorphic within a single species, and they're very variable. And so phasing the genome allows us to really interrogate some of the differences that are present within these and some of the polymorphisms that are present. So using this pipeline, we have been able to, for the first time, annotate the NLRs in a phased plant genome. And one of the first things that you'll probably see is that there is quite a big difference between the number of NLR genes that are present in the different chromosomes within a single haplotype. So for example, chromosome 5 is very gene rich, as are 2, 3, 7 and 8. And this is similar to what we see in other Myrtaceae genomes, such as the Eucalyptus grandis genome. What was really interesting to us, and not exactly surprising, but we do see a variation in the number of these genes between haplotypes as well. So we can see that between haplotype A and B, we do see up to about 30 gene numbers being different between those chromosomes. When we take a little bit of a look further into the types of those NLRs that are present, so in blue we have the ones that contain this TAR region, and in the purple we have the ones that contain the CC or the coil-coil domain. We do see as well that some of the chromosomes are more rich in some of these types of genes, and this is really due to the tandem duplication events that occur to expand this gene family as well. So why this is all important to us and why we're interested in looking at this is because for some NLRs in different species that are known to confer resistance to particular pathogens, there is evidence for allelic series being there. So there's variation and allelic differences in the NLRs, and we're really interested in understanding if that same thing is occurring within the Melaleuca quinquenervia genome. So just to go into a little bit of the integrated domains, so I alluded to these earlier, but these can act as decoys for pathogen effectors. So when the pathogen is releasing effectors into the host, they have targets on the genome. These integrated domains will generally act as decoys for those effectors, which allows for the resistance genes to detect the pathogen before it's actually bound to the actual target and allow for a response to that. So we found that up to 6.8% of the NLRs in the Melaleuca quinquenervia genome contained these integrated domains, and there was slight variation in the number of NLRs that contained integrated domains, but what was most interesting was the diversity that existed between the different haplotypes. So haplotype A and haplotype B contained different types and different numbers of those integrated domains. The most common domains were the Werke transcription factor domains and the zinc finger bed domains. And we're particularly interested in these because, for example, the zinc finger bed domains have been found in NLR genes that confer resistance to rust in wheat and barley. So the next steps in this model system is to screen the progeny from the tree that we used to generate the high quality phased genome. So we collected seed from the parent tree that we used to generate the genome, and there was a large subset of those that were grown from seed and inoculated to screen them for their response to the pathogen. Interestingly, even though we had a highly resistant tree, we did see this variable response from highly resistant, which is typified by the hypersensitive response, where you can see the localized cell death, where the host has responded to the pathogen, all the way through to highly susceptible, where we can see that the host hasn't responded at all, and we don't see any localized cell death there. So what I'm really interested in is conducting some transcriptomic analysis to look at differential gene expression between the highly resistant and the highly susceptible hosts. I have done this actually by using naive hosts. So using the initial subset of the progeny, I looked at the phenotypic ratios for each of these different phenotypes and used this to determine how many plants I would need to collect from to be able to get enough of these for transcriptomic analysis. So we're aiming to identify some of the genes that are involved in resistance or the resistance pathways, and this data will be available as of July next year. And it's being funded by in a partnership with the University of Sydney and also the Department of Primary Industries here in New South Wales. So now just a bit of an introduction into how we intend to apply all of these methods to different Melaleuca species. So in selecting the different species to screen, I wanted to identify species that were potentially at risk of Myrtle Rust, but hadn't yet been exposed to the pathogen before. So Western Australia is a biodiversity hotspot. The southwest region of Western Australia is a biodiversity hotspot, and it's rich in Myrtaceae species. Unfortunately, many of the species that are endemic to this area are already under threat due to other pathogens such as Phytophthora cinnamomi, which is the causative agent of jarrah dieback. And climatic mapping has predicted that this region will be suitable for Myrtle Rust. Unfortunately, as of July this year, Myrtle Rust was detected for the first time in Western Australia in the Kimberley region of the state, which is next to the Northern Territory. So it's believed that the pathogen spread from the Northern Territory into Western Australia, and it was detected on 2 Melaleuca species here. Unfortunately, due to how widespread this region is and how hard it is to actually get up there, the extent of the spread is unknown, and we also don't know what species that are endemic to these regions are susceptible to the pathogen as well. So this part of the project really focused on screening Melaleuca species from WA that are in these high priority areas, so the southwest region and also that northern Kimberley region. So to do this, I sourced seeds from Melaleuca species from the seed banks in Western Australia. I've grown some of them and inoculated them under controlled conditions with the pandemic strain of Myrtle Rust. I've been monitoring them for their response to infection and scoring them from highly resistant to highly susceptible, as we saw in that scoring system earlier. And then the downstream work will be to screen the resistant seedlings for genes that are related to resistance that we have identified in Melaleuca quinquenervia. So I have sourced seed from 25 different species. 8 of these are from the Kimberley region and 17 are from the southwest region. Just due to some difficulties with getting them to germinate, even though I said it was very easy. We've had some problems with our glasshouse here at the University of Sydney, so it's been a little bit challenging. But of the 5 species screened to date, many of them are showing high susceptibility to the pathogen, all the way up to about 86% of the plants screened being highly susceptible. So this was of particular concern, as some of those species didn't even show this variable response to the pathogen. For example, the Melaleuca wanneroo, which you can see here. Of the 80 plants, 86% of those were scored as score 5, so highly susceptible. And the ones that did not show any symptoms were unsure whether or not that was just due to the material not being at the right age to become infected, or whether or not that was actually resistant. So there will be some follow-on screening from this to determine and confirm all of these ratios as well. So from a conservation standpoint, this work is very important, as it's going to allow us to determine which species within these regions are highly susceptible to the pathogen and allow for seed to be collected for conservation efforts. It'll also allow us to identify where these species are within Western Australia, and they can be monitored. So if we are looking for any incursions of Myrtle Rust into Western Australia, these species can be monitored, as they might be able to be used as an indicator of the pathogen entering the state. So how this all ties together, the future work is going to be to take the transcriptomic data that has been generated in the first stage of this project on the highly susceptible and highly resistant Melaleuca quinquenervia, and by identifying the genes and pathways involved in resistance to Myrtle Rust in this one species, we hope to then be able to screen our Western Australian species for these same genes and pathways. So it's really to validate and to test the utility of using Melaleuca quinquenervia as a model for genetic research. And I've recently undertaken a nanopore sequencing workshop at the Australian National University with the aim to be able to generate some whole genomes for these susceptible West Australian species as well. So with that being said, there's a lot of people to thank. So obviously my PhD supervisors, all of the collaborators on the genome project, the technical staff at the University of Sydney, which it wouldn't be possible to do without them, the staff at the Department of Biodiversity, Conservation and Attractions, who have been involved in providing the seed for all of the West Australian species screening. Robert Mackinson as well, I would just like to thank for putting me in touch with everyone in Western Australia about the species screening, and also all of our funding partners as well. So before I say goodbye, I would just like to get a little bit of feedback from anyone in the audience who may be familiar with this area. But I have received a fellowship from Eucalypt Australia, funding a citizen science project to monitor the spread of Myrtle Rust across Australia. So compared to how this has worked in New Zealand, we only have 129 public observations in Australia, and I think when I looked at the Myrtle Rust reporter yesterday, it was up over 2,000 in New Zealand. So the project is really aiming to spread awareness of Myrtle Rust, the host, the symptoms and the impacts to the citizens around Australia, to raise awareness about it. And the data will also be used for monitoring the spread in Australia and identifying hosts. So it's being launched in 2023, but if there's anyone online that has been involved with the Myrtle Rust reporter in New Zealand or has any advice for me, that would be most appreciated. And I think Renee will be sharing my email address later. Okay, I think that is it from me. I'll hand back to Renee.
Thank you very much, Alyssa. Amazing to see these results coming out when you're only, I guess, roughly halfway through your PhD. And yeah, excellent to see that you're interested in adopting iNaturalist for reporting. So something we can certainly chat about a little bit later. I was made aware the other day that for iNaturalist New Zealand, at least, Austropuccinia psidii, the causal agent of Myrtle Rust, is now the most commonly reported fungus on iNaturalist in New Zealand. And we did see over 800 observations in the year ending the middle of this year. So that was mainly throughout the last season. So I do have a couple of questions for you, and I would encourage the audience to get any questions they have in the question box. So great to see that you're going to be expanding this work to other Melaleuca species. It's a bit of a tongue twister, isn't it, that Melaleuca? Especially when you add in the quinquenervia. I probably should have practiced that a little more. How many of those species are there in total in Australia, just for those of us who aren't familiar? And I'm also wondering how many of them are threatened from other causes than Myrtle Rust.
Yeah, so on the exact numbers, I'm not actually sure, to be completely honest. So I think Melaleuca is the third largest genus in Myrtaceae. And we do see certain species, for example, in that southwest region of Western Australia, Phytophthora I mean, generally in Western Australia, Phytophthora cinnamoma is a large issue. So I think if anything, even if these species aren't currently threatened, if anything we've learned from what we've seen in Australia and some species becoming threatened as a direct result of myrtle rust is that we need to focus as broad as we can. And so I was really focusing more on the species that are present within these areas that are showing climatic suitability to the pathogen, rather than looking at the ones that are already threatened.
It sounds like an
Hope that makes
entirely
sense.
sensible way. And we do have a comment here that land clearing is one of the biggest impacts on Melaleuca species. So thanks for that contribution from the audience. So just one more question from me, because we do have one there from the audience, which is great. So Manuka or Leptospermum scoparium has resistance and susceptibility in leaves as well as stems, complicating, I understand, attempts to study resistance in that species. I'm just wondering if that's also the case for any of your Melaleuca species, or is it a bit more straightforward? It's just the leaves that are really impacted with that
So
species.
we see stems impacted as well. My focus is on the leaves for now, but it definitely is something to consider. So in doing this scoring, I have refined the scoring system a little bit to suit Melaleuca, because what we were seeing is that we might be able to score at score 4 and score 5, but we also identified some of those where the stems were resistant or the stems were susceptible. So there is that added layer of complexity in there. For now, we're just focusing on the leaves, and I think in seeing some of those images that I showed sort of towards the end of my talk as well, you could see some of the other WA species that the stems are infected. So it would be something that we could definitely expand on in the future as well.
That sounds like a good idea. All right, a question that we do have from the audience. Have you considered investigating mechanisms of quantitative rather than qualitative resistance? And could quantitative mechanisms be more durable?
Yeah, so I'm a little bit unclear on that question, but I think it's more around the scoring, I think, in how much of the fungus we're actually seeing is there. So I did consider this very early on. The initial plan was to extract DNA and RNA simultaneously from these plants, but unfortunately a triazole reaction does not work for them. I discovered very early on, so I just had to use a kit for RNA extraction. But I think it's something that we can also expand on, because everything we're currently doing is based on qualitative, and it's just what we're seeing rather than actually how much of the fungal load is there. So again, definitely something that we are considering adding in the future, and it has already been considered. It's just, yeah, the methods didn't work as expected, unfortunately.
Indeed, and of course this is one of the tremendous challenges of science, these kinds of technical difficulties that can be rather hidden behind the scenes, and even seemingly simple things like keeping your greenhouses at the required temperature.
Yes.
And
watering on.
yes, it can be very challenging, and certainly something that researchers working in the Myrtle Rust space in New Zealand have been challenged by as well. All right, a few more questions coming in now, which is great. Are there races of the rust?
Yes, so currently in Australia, we believe we just have the pandemic strain here. So for all of the controlled inoculations, we are using the pandemic strain. So all of the screening that you saw was based on that. I guess this is one of the reasons as well for getting this citizen science project set up, is to actually be able to do some targeted collections around Australia. So by providing the locations of the rust around Australia. We're hoping that somebody will be, there's no funding for it at the moment, but we're hoping that somebody would be going around and actually collecting samples to be testing for any differences or any changes in the pathogen as well. But as far as we're aware, even with the West Australian incursion, there was some work to determine if that was a new incursion or if it was still the pandemic strain. And some researchers at ANU, and I think in collaboration with the University of Queensland, were able to show it was still the pandemic strain that was there.
We have another question that ties in quite nicely with that. Do you believe the presence of myrtle rust in the north of Western Australia significantly increases the risk of it spreading to Western Australia's southwest?
Yeah, so I guess there's a couple of ways it can go. One concern is it spreading down the coast of Western Australia. Probably what we're more concerned about now is spread along the south coast. So spores have been detected in traps in South Australia, and while we haven't seen any disease symptoms there, if the spores are making their way across by wind, then that's probably more of concern. So Western Australia is, as you saw in the map earlier, very large, and so there are different climatic conditions across there as well. So we would think that the Kimberley region being very hot and humid is conducive to the pathogen, but that central region, there's a lot of desert, so obviously it can spread by wind, but yeah, so there's really a couple of ways that it could get there, and I think implementing some spore traps. I know that I'm at Pests as an example, or BioScout as well, which is using machine learning. They're, I think, currently training their models for Myrtle Rust. Something like that would be highly beneficial to start implementing even at the borders, just to see if we are getting any spores coming across either way.
It's very interesting to hear about that spore trapping work. That is not something I've heard about before for Myrtle Rust. So the organisations carrying that out are the two that you've just mentioned?
Yeah. So I'm at Pests. They have some sentinels. They have been set up, I think, in some different areas, more along the east coast, and there might be one in South Australia as well. And I'm fairly certain that that's the one that detected the spores in South Australia. And then there's BioScout, which is a startup based here in Sydney, and they're hoping to train their models for Myrtle Rust as well.
Excellent. All right. And one more question, a little bit more technical now. Nice work. Will you be searching for genes and pathways other than the NLR gene family?
Yes. So I think we understand that they have a very important role in resistance, but from all of the transcriptomic analysis to date, they actually don't come up all that often. And I think that's partly to do with there already being sort of a base level of expression there of these NLR genes. And so we don't always see them being upregulated when there's exposure to the pathogen. So this is part of the reason that I have conducted this work on naive hosts, which was a very painful experiment, I might add. There are a lot of collections, but we're really hoping that by analysing plants that haven't been exposed to any pathogens before, that we might actually see some differential expression that might be lost when we're looking at hosts that have already been exposed to the pathogen. But this is definitely expanding beyond the NLR genes. So the transcriptomic analysis will just identify all of the differentially expressed genes to then determine what some of those genes and pathways are.
That sounds excellent. And I see another webinar in that. Any luck, we'll be able to convince you to come back.
December 23.
That's right. You could bring us into Christmas next year. And you've got some more fabulous results coming through. All right, well, that's it for questions from the audience. And indeed, Christmas is coming. So I will grab my notes and wrap this one up. Thank you very much. Really excellent work. Now, a video of today's webinar will be made available on the Beyond Myrtle Rust website in the next couple of days, and it will also be emailed to everyone who registered. The next webinar will be on Wednesday, February 15th from a yet-to-be announced speaker. So look out for the invitation in the new year to see who that is. I do have a number of excellent candidates, so fear not. I'm sure one of them shall deliver another great webinar for us. Thanks to everyone out there who has supported another great year for the webinar series by presenting or by coming along. And I look forward to bringing you another excellent lineup of speakers next year. Have a great break, everyone. Haere rā, and see you next time.